Hi everyone, my name is Boon Hoa. I'm the Regional Marketing Specialist from GenScript and I am your moderator for today. GenScript is the world's leading biotech company providing life science services and products with gene synthesis, peptide, protein, antibody, and preclinical drug development service capabilities. In today's webinar, Dr. Edward is going to introduce the state of art in antibody engineering for better therapeutic antibodies. Dr. Edward Wong is field application scientist from GenScript, and he completed his PhD degree from the University of Adelaide. He worked as a postdoctoral fellow at the National Cancer Centre in Singapore for 3 years, where he successfully published his findings on the predictive factors of genetic screening of BRCA1 and BRCA2. He also published his works on the importance of genetic testing of 25 breast predisposition genes using NGS technology. Both of his works were reported in PLOS ONE and MPJ Genomic Medicines. So this webinar will take around 40 minutes and it will be a recorded session. You can type in your questions in the Q&A box and we will answer them after the presentations. If we didn't get to answer your question during the Q&A sessions, we'll be sure to take down and email back to you. So over to you right now, Dr. Edward.
Thank you for the introductions. So good day, everyone. My name is Edward. It's a great pleasure to be invited on this webinar series. And in this webinar, I am going to discuss about how we can engineer the antibody in order to improve the properties using the synthetic biology approach. Now, let me walk you through to the contents of my presentation today. I will begin with the introduction to antibody engineering using the synthetic biology approach, followed by sharing a couple of case studies to demonstrate how GenScript could have helped to rapidly identify and validate the antibodies repertoire using the SARS-CoV-2 virus antibodies as example, and then to improve the antibody affinity using the synthetic biology approach. Now, without further delay, I shall begin with the introduction. Okay. With the advent of biologics, the researchers can now reach the targets that previously known as undruggable. However, these biologics are still relatively larger and more complex than the small molecules. And also there lies a number of challenges for the scientists to create and produce these biologics for therapeutic purpose. So if you look at the discovery and development workflow for the therapeutic antibody, we can see the possibility to encounter several challenges along the way. For example, during the lead generations, we might miss the good binders or not enough binders being generated. During the lead optimizations, difficulties arise with the poor library coverage or the library bias. And when produced the optimized antibody, we can suffer from the low yield and facing the scalability issue. And lastly, the scientists are still left with the questions of how to create and produce the complex antibodies with the better target and also be clinically effective. So in order to answer these questions, the scientists have recognized that we need to think the biology differently these days. That means to say that we need to think like engineers. So this was how the synthetic biology was born. So here I show you an overview with the use of a synthetic biology approach, including the protein engineering in the diversified biotechnological and the biomedical science field. So for biomedical science applications, that includes the developments of the antibodies, the designs of the drugs for the protein scaffold, as well as the drugs for the different diseases. They include the diabetes, the cardiovascular diseases, the modulations of the enzymes and cytokines for therapeutic purpose. And on the other hand, The synthetic biology approach has also been used to improve the activity of the enzymes, the specificity, the stability, and so on, in order for the industrial usage. The high expression of the catalytic enzymes under the stress conditions for the environmental applications. In addition, the synthetic biology has also been used to improve the biomaterials, such as the vector engineering. And last but not least, the synthetic biology has also extended its arms towards the nanotechnologies and also for improved biosensors. Now, to understand how it is done, we must know what is synthetic biology. When I look for the information, I came across the article that published in the Chemical and Engineering News in the year of 2000, in which the synthetic biology was defined as the use of the synthetic capability of organic and biological chemistry to design the non-natural synthetic molecules that has the function in biological systems. And as the time goes on, the definitions has become broader, which is shown here: the design and constructions of a new biological parts, devices, and systems. And those parts, devices, and systems are really the words that come from the engineering. So one of the ways we start to think the use of synthetic biology to redesign of the existing natural biological systems for the useful purpose. As you can see the picture here, through the biodesign process that includes the design, build, test, and deployed, it can offer the possibility to offer the new solutions for the global healthcare, such as the productions of a high values of the chemicals and drugs. Also, to improve the novel bio-based materials in manufacturing, as well as to improve the biomaterials in order to overcome the environmental challenges. So here, I would like to briefly discuss about how we could apply the engineering principle to biological systems. By design, for an example, I want to build a machine that gives me functions, and I know how to draw a way to get there. The modeling is all about the mathematics. That means that we can come up with an equations to support the design. And the characterizations actually is the practice to going through from the design to the point where you build at. And through the testing process, you characterize the system as a whole, as well as the individual part. The abstractions really means actually being to take now a larger view. And if I go back to the definition of the parts and devices, I need not to go into very details of the information, but knowing that I want a bigger functions, and I can encode in a simple way. So the key technology for synthetic biology is the DNA synthesis. And with the reductions in the cost of the DNA sequencing and DNA synthesis these days, we can access to a more detailed genetic information and a more powerful tool for genetic engineering than ever before. So this is a simplified diagram that shows that the engineering begins with the definitions of the sequence in the natural host, and then you can reconstruct of those to give you a piece of the synthetic DNA. And you can have this piece of a synthetic DNA to give you a functions that allows you to convert the input into the output that you want. Or you can stringing these devices together, that one has one function and another has a different functions, and compile these devices to give you the features or the structures of interest. So at GenScript, we have three core platforms to support the drug and discovery research. Our molecular biology service is powered by the next generation of DNA synthesis technologies that allows us to synthesize any genes and to clone into any of the vectors of interest. We have 17 years of experience and had completed more than 600,000 of the gene synthesis projects. And what I would like to highlight here today is we have more than 10 years of the experience in mutant library constructions to create the libraries with unbeatable control and also a complete coverage. For protein service, we have a high density of the protein expression systems that allows the increase for the recombinant antibody expressions from 4 to 8-fold, and also the increase in the protein's expressions up to 100-folds. And lastly, our award-winning antibody service had completed more than 150,000 antibodies to date, and also offers highly customizables and being able to tailor for your research needs. So today, I am going to share with you how GenScript incorporated these 3 services to help in the drug discovery and the development process. And along the way, I will highlight some of the cutting-edge technologies for the lead validations and the lead optimization. And now let's move on to discuss about how are we using our technologies for the lead identifications and validations for to discover the SARS-CoV-2 antibodies repertoire. So as you know, the assay development plays an important role during the early stage of the drug discovery process. The recent outbreak of the coronavirus in December 2019 has demanded a great need in quickly generated a high quality of the monoclonal antibodies in order to develop a highly sensitive and reliable immunoassay, such as the immunohistochemistry, the ELISA-based immunoassay, the Western blot, flow cytometry, and so on. So for this reason, we coupled the unique advantages of the rabbit immune system and the cutting-edge Bacon single B-cell screening technology to maximize the opportunity to rapidly identify the high quality and diverse monoclonal antibodies towards the spike RBD proteins. So with this technology, we completed the immune cells isolations and enrichments, the single cell sorting, as well as the on-chip bindings and also the blocking screening within only 24 hours. And this process, as you know, that would have taken months if we rely on the traditional ELISA-based hybridoma technologies and also the synthetic DNA library antibody screening methods. So with these technologies, altogether, near 15,000 of the single B cells were subjected to automated screening using an antigen-specific bit-based assay to identify the monoclonals that positive for the RBD bindings that were being secreted by its correspondent B cells. And in total, the antibodies screening and selections resulted in 142 antigens positive monoclonal antibodies hits. So the evaluations of the top rabbit monoclonal antibodies using the BioCore SPR platform revealed that the rabbit monoclonal antibodies that are discovered by the Bacon technologies possess a high affinity compared to the mouse monoclonal antibodies using the hybridoma technology. Most of the rabbit monoclonal antibodies displays a 10 to 100 folds of the high affinity compared to the top clones of the mouse monoclonal antibodies as highlighted in the red boxes. So these rabbit monoclonal antibodies were then further analyzed with the epitope binning and found that the rabbit monoclonal antibodies that discovered from the Bacon platform were distributed across 4 different epitopes. In contrast, using the hybridoma approach, only 1 epitope was occupied among several rabbit monoclonal antibodies. So this was actually in agreement with what the broader scientific community has observed. That is a more diverse antibody repertoire can be discovered using the Beacon platform at an efficiency that cannot be achieved using the hybridoma technology. And the generations of the diverse monoclonal antibodies to recognize different epitopes on the target proteins are critical for a variety of reasons. The sensitive and reliable immunoassay development relies on the availability of a variety pair of the monoclonal antibodies so that the researchers can evaluate in-house to identify the quality of the top candidates that best works in their assay. In addition, the specialized immunoassays such as the sandwich ELISA relies on the antibody diversity due to the need of the monoclonal antibodies pair that recognize and bind to the unique epitopes on an antigens. In fact, from the spike RBD monoclonal antibodies that we identified, we developed a highly sensitive sandwich ELISA using the clones BS-R2B2 and also the BS-R2B12 biotinylated clones with a sensitivity for the spike RBD proteins at 20 picograms per mil. So with these technologies, in addition to the antibodies that presented in the case study, we have developed over 80 SARS-CoV-2 control antibodies which are available on demand. And we have a wide range of the nucleocapsid proteins monoclonal antibodies and the spike proteins monoclonal antibodies. For spike proteins, we have the monoclonal antibodies that recognize only to the spike S1 RBD, and some of the monoclonal antibodies possess the neutralizing functions. Additionally, we also have the monoclonal antibodies that recognize only to the spike S1 and S2 ECD. Furthermore, we have developed the assay cell lines with the high expressions of the human ACE2 receptor. So these cell lines include the HEC293T, A549s, the HeLa, as well as the CHOK1 cell lines for drug discovery purpose. For example, these cell lines can be used for neutralization assays in order to screen for the antibodies and other small molecules that can block the pseudovirus or the live virus infections. Additionally, they can be used for affinity analysis of antibody or other molecules to the spike proteins. And finally, they can be used for investigating the SARS-CoV-2 infections mechanism. So next, I would like to switch gear and discuss about the lead optimizations. So as we know that the monoclonal antibodies are being used in the diverse therapeutics and diagnostic applications due to several of their attractive properties, such as the high binding affinity and also the specific mediated by the CDR within the variable regions, the high conformations, stability, and solubility that allows the formulations and extended storage of the concentrated antibody solutions with minimal aggregations, and also the relatively ease of manufacturing different monoclonal antibodies using a similar platform process. So that enable the generation of a large number of the lead antibodies. However, most of the antibodies that are identified during the initial discovery process are not suitable for therapeutic use due to a couple of several reasons, such as the binding affinities are not high enough, or the optimized properties can lead to the defects in other properties. So the main challenge to the researchers are, how can we improve the antibody for therapeutic purpose, and how can we improve the antibodies without compromising other properties. So fortunately, due to the increased amount of the experiments, the protein engineers and the scientists have developed some kind of the uniform lead optimization strategies. So the first strategies that I would like to introduce today is known as the rational design. I believe that many of you are familiar with this strategy. It is an in silico approach that requires information on the amino acid sequence, three-dimensional structures, and also the antibodies database. So with this, the researchers are able to identify the hotspot rapidly that associated with its property. And following to that, the scientists can then introduce the variant into this hotspot through the site-directed mutagenesis. As mentioned, the advantage of this strategy is it allows the researchers to rapidly identify the hotspot that associated with its properties, and thus restricted the number of mutations that require to be constructed and to be screened. But the drawback of this strategy is the detailed structural knowledge of an antibodies are not always available. Even if it is available, it is always very difficult to predict the effects of various mutations. So the second lead optimization strategy is known as the directed evolutions. This strategy has been proven as a powerful tool for tailoring either the enzymes or antibodies for the past 2 decades. Fundamentally, it mimics the natural evolution process in vitro under a properly controllable selective pressure for targeted purpose. So the key principle of this strategy is to create a molecularly diverse library to screen for the variants with the improved performance. And this molecularly diverse library can be created through the random mutagenesis approaches, including the error-prone PCR and the DNA shuffling strategy. The advantage of this strategy is it allows the antibody modifications in a situation where the information regarding the structure and the mechanisms of the target antibody is unknown. And to obtain the expected level of the optimizations, several rounds of the evolutions are required and to screen a large number of the variants. So you could have imagined that the entire workflow of the directed evolutions is time-consuming and laborious. Additionally, it requires a high-throughput screening methodology. So based on these 2 strategies, you could have realized that it is very important to have a well-constructed mutant library that can be very useful for lead optimization. This I show you the lead optimizations workflow. You start off with the genes of interest, and then you construct a deep mutation library, which you can then express in the host, and then to screen for the optimal variant. You can use that knowledge to construct another mutant library, which you can continue to screen and isolate for a better variant. And you continue this cycle several rounds until you have your optimal lead candidate. So because the optimization process can require multiple rounds of the library constructions and screening, it is crucial that your library contains all of the sequence that you want to test. And these sequences are represented in equal amounts to reduce the overall screening and lead to the optimal sequence quicker. So the question is, how are these libraries constructed? So to generate the mutant library, many of the researchers would probably use the in-house methods such as the degenerative mutant library, NNK or NNS. But this randomization library are bound with the disadvantages, including the researchers unable to control the amino acid ratio that incorporated in each of the position. And when you couple the nucleotides, each of the nucleotides will have different coupling efficiency. And because of that, you indirectly introduce the bias into the library. So you will see the A, for example, if you have A to C has a higher efficiency, you will probably see a lot of the C's in your library. So this is something that the scientists are able to control. What all that means is the researchers is making the library that is out of their control in terms of the design, so that leading to the presence of the unwanted species in the library, and will make them more time to screen for the variants. So in order to overcome these limitations, at GenScript, we developed our proprietary technologies that allows the researchers to use all 64 codons when creating the mutant libraries while avoiding the stop codons, and also to allow the codon optimizations, and ultimately to eliminating the sequence bias to create a mutant library that is meaningful and also at an equal representations, and therefore expediting the downstream screening. So we call these technologies the precision mutant library. Now, I will walk you through the approach that GenScript utilized based on our semiconductor technology to construct different types of the libraries for different purposes. So we use this semiconductor chip technology to synthesize the oligo that contains only the particular mutated sites on the sites of interest. And then we stitch it together to the constant regions using our proprietary assembly method. With this, it allows us to generate a pool with large number of different variants. And based on this technology, it allows GenScript to construct 3 different types of the libraries. The first type of the library is known as the site saturation library, where we can introduce a particular mutation onto the specific site, and then a saturation scanning libraries where you can have all of the mutations across the regions of interest, or in the scenario where the researchers would like to mutate different sites simultaneously in a combination manner, our combinatorial mutant library can fulfill the demand. So the final end products will be delivered either in the PCR products, or we can help to clone these variants into the vectors of interest using our proprietary cloning strategy. So, and these variants will be validated using the NGS sequencing in order to guarantee the coverage as well as the accuracies that are being generated. So now I would like to show you a case study to demonstrate how the PML service will have helped to reduce the screening burden while obtaining optimized antibody candidate quicker. So in this case, we created a saturation mutagenesis library of 63 sites across 6 CDR regions in both light chain and heavy chains. So each site was mutated to all 19 non-wild type amino acids. For library construction workflow, as I mentioned earlier, we use our semiconductor chip technology to synthesize the pool of oligos that contain only the particular mutations on the site of interest, and then we stitch it back together with the constant regions. Then each of these synthesized variants was cloned into the plasmid and then pooled together. Here is the NGS result showing the ratio of the amino acid at each mutated site. Each bar here indicates individual sites, and each of the colors represents an amino acid. The thickness of each color band is the percentage of each of the variants with the particular amino acid at that site. And from this data, we observe an equal distribution of the amino acids across 63 sites, with the variants representing all 19 non-wild type amino acids present at each site. The next questions would be, can we really use these libraries to find the mutant antibody candidates with improved affinity? So to answer these questions, a total of 3,000 clones arising from the precision mutant library containing the 63 mutated sites were screened for candidates with improved affinities against the target antigens with a cutting-edge technology known as FASIBA. So using this screening method, we successfully identified several mutants with significantly improved in affinities as shown in the blue circle here. So these mutant clones with a significantly improved affinity from the placebo screening were further characterized for their binding affinities using the BioCore SPR platform. So the SPR assay results revealed 5 mutants with the affinities higher than the wild type. And one of these mutants displays an affinity even in the femtomolar range, as shown in the red boxes, highlighting the capability of our pre-ML to generate such a high affinities mutant sequence. So in order to assess the combination effect of the 5 beneficial mutations and potentially further improve the binding affinity, a combinatorial library that allows for the introductions of several mutations simultaneously within each single mutant was constructed using the same semiconductor-based DNA synthesis technology. So a total of 96 clones from the library transformations were subsequently screened using the FASIBA, as I mentioned, and the characterizations of a number of top clones from the FASIBA screening through the SPR binding assays reveal a few combinations mutants with improved affinities as shown in the table here, compared to their corresponding parental clones. So this result highlights the ability to utilize different types of the precision mutant libraries to enable the generations of the improved high affinity binders. On the other hand, the degenerative mutant library with the NNK approach was constructed for comparison's purpose. And if you look at the distributions of the NNK library, we were unable to achieve the equal distributions of the variants across the mutated sites, with certain amino acids being more represented or being underrepresented than the others. Additionally, based on the side-by-side comparisons between the NNK and the PML libraries, we were able to achieve a high diversity by screening less colonies when using the precision mutant library. So these results show that with PML, it allows us to create a library with better coverage and distributions compared to the traditional approaches, and reduce the number of required clones to screen, and therefore improving the overall screening efficiency. So before I wrap up my presentation today, I would like to highlight that with our precision mutant library, we are able to have a precise control over the codon usage and allow the user-defined amino acid distributions within each variant. And this allows for the construction of the mutant of a superior library diversity with the guarantee of more than 90% of the coverage. So here I would like to thank you for your attention, and I'm willing to take in any questions for discussion. Thank you.
All right, thank you so much, Dr. Edward, for your wonderful presentations. Now we will start to answer some questions from the floor. All right, so in both the protein engineering strategies, you have specified the disadvantageous points. So I was wondering if those points are crucial in these strategies?
Yes. So as I mentioned that, in the real world, as I mentioned, we need to generate a large diversity of a high quality of the antibodies quicker, so that we can develop the immunoassays quickly as well. So the strategies that I mentioned, the two strategies that I mentioned earlier, would actually takes a lot of time, like I mentioned, especially the directed evolution strategies. So for these strategies, it needs to take time to construct a large molecularly diverse libraries And so, and also it will take a lot of times for the researchers to screen for the variant, a large number of the variants with the improved performance. So in terms of the times, as well as the efforts-wise, it is incompetent compared to the new technologies that I presented, which is the use of our Precision's Mutant library. Yeah.
All right, thank you. So the next question is, I have heard about a new immunoassay innovations called Open Sandwich ELISA that enables us to reduce the washing step and incubation period. From Open Sandwich ELISA, do you know how to test the compatibility of the chimeric receptor based on the response from many antibodies in the chimeric receptor?
Well, I apologize that I'm not an expert in this Open Sandwich ELISA, and maybe I can take down your questions and get our scientists to to reply you after this webinar.
Okay, sure. Thank you, Dr. Edward. So the next questions will be more concerned to the logistic and the transport issues. I would like to ask, if we synthesize the peptide in GenScript, will there be any transportation issue to Sabah, Malaysia, with the current COVID-19 situations?
Okay, so for these logistic issues, for informations, GenScript had actually set up the logistic hub in Singapore. So I believe that now after the peptides be synthesized, the peptides can be delivered directly from the China to Singapore, and from Singapore, we can arrange for the logistics to deliver this peptide to Sabah. It shall not be an issue, because this is how we usually done for the customer from Malaysia side.
Great. So the next question is, do you have a limit of the library size, and what is the maximum library size?
Yes, this is actually a great question. We do have the limits in the library size that we construct. For the cloned libraries, the library size will be dependent on several aspects, including the copy number of the plasmid of your choice, and also the size of your final construct. So generally, we are able to achieve a library of approximately 10 to the power of 6 to 10 to the power of 7 using a high copy number of the plasmid. So when necessarily, we can try to scale up from 10 to the power of 7 to 10 to the power of 8. But this is the maximums that we can achieve so far. However, on the other hand, if the clients prefer the PCR as the final end product, the maximum library size that we can reach, as far as I know, is 10 to the power of 11.
Okay, thanks, Dr. Edward. The next question is, what is the percentage of coverage will you achieve?
The percentage, okay. So in general, we can ensure to achieve a greater than 90% of the coverage, as I mentioned, using the validated using the NGS. So our existing clients are actually quite satisfied with what we could have achieved so far, and this is actually what we are proud of, using our sophisticated systems and a great depth of the experts team in GenScript. But as shown in my case studies, we are trying our best to achieve 100% of the coverage. It depends on the project. So my advice to the audience is, if you have a research project, would like to utilize our Precision's Mutant library service, we hope that we could actually arrange a teleconference or for the sit down together with the clients and understand about the project needs so that we can give a better advice in terms of their how we could help the researchers to synthesize the variants and also to achieve the coverage that they want. Thank you.
Great. Thank you, Dr. Edward. So the next questions will be, if I would like to mutate only CDR3 for screening the good antibodies, do you have any idea with only this mutate region?
Meaning that does the client means we help them to design the CDR regions, or we just help them to synthesize? So for these questions, right, I would like to highlight that in terms of the designs of the mutations for the variants in the CDR3 regions, we usually rely on clients to have to have this design to be ready, and then where GenScript comes in the we will actually go through the variants that the clients actually design, and based on our technologies, we will advise and see whether or not it's actually synthesizable or doable using our in-house technologies. So this is how the entire service that we are offering.
Okay, thank you, Dr. Edward. The next question is, when you mention the format of PCR products, do you mean it will be the mixture of purified PCR fragments of the antibody?
Yes, that is correct. So it's not an antibody. It's actually the PCR purified PCR products of that oligos with the contents the particular mutations on that site. And then after that, once the researchers receive this mixed pool, they themselves will need to clone into the vectors of interest.
Great. The next question is, what are the limitations in the combinatorial mutant libraries in terms of the total number of sites and total number of mutations?
This is a very difficult questions here, because the thing is like the limitations is actually depending on several factors, including the scattering patterns of the mutation sites and also the mutation pattern in each site. So for the combinatorial mutants libraries, unlike the site saturations and also the saturations mutagenesis library, our experts really need to evaluate the projects in order to give a precise library design and answer to these questions.
Okay, great. Thank you. So I have no more questions from the floor. So to all the attendees, if you have any questions regarding our presentation just now, please feel free to type in the Q&A box. So, yeah. And if there is no questions, I would like to thank you once again, Dr. Edward, for your great presentations. And thanks all of you who joined this webinar today. We have come to the end of this webinar, and we hope you find this webinar useful and beneficial for your work. So yeah, there's no more questions. So thanks everyone, and we will close this webinar now, and we hope to see you again. Thank you, Dr. Edward, for your presentations. Thank you, everyone.
Thank you.